Let's microbiology it by micro-mind mapping. This will allow you to retain the basic information before you open your textbook or go to lecture. Our first core is cell comparison. Stemming from that, there is prokaryotes. Now here's all of the information for prokaryotes that I want you to know. 1, there's no nucleus. However, there is a nucleoid region. The second one is that prokaryotes have circular DNA. They have no organelles. They have peptide or glycan wall. And how do they divide? By binary fission. Now going back to our cell comparison, our core, stemming from that, we have eukaryotes. And here's some information that I want you to know about eukaryotes. They have a nucleus. They have multiple chromosomes. They have membrane-bound organelles. They have chemically simple walls. Most of them do. And they divide by mitosis. So I want you to commit this to memory, and we'll go on to the next section. Prokaryotic and eukaryotic cells represent two fundamentally different organizational strategies of life. Prokaryotes include bacteria and archaea, lacking membrane-bound nucleus and membrane-enclosed organelles. Their DNA exists as a single circular chromosome located in the nucleoid region, and cell division occurs through binary fission. The absence of histones and compartmentalization allows for rapid replication and metabolic flexibility, which contributes to their evolutionary success in diverse environments. In contrast, eukaryotic cells possess a true nucleus containing multiple linear chromosomes associated with histone proteins. They contain membrane-bound organelles that compartmentalize metabolic functions, allowing for increased cellular complexity and specialization. Eukaryotic cells divide via mitosis, ensuring accurate chromosome duplication and segregation. Cell wall composition further distinguishes these groups. Prokaryotic cell walls typically contain peptidoglycan, while eukaryotic cell walls, when present, are chemically simpler and vary among organisms, such as cellulose in algae and chitin in fungi. Some eukaryotes, like protozoa, lack a cell wall entirely and instead possess a pellicle for structural support. Which of the following is false about fimbriae? You need to know what fimbriae are. And they're short appendages that are for attachment or adhesion. So we're going to go down the list. A, they are composed of protein. B, They may be used for attachment. That's true. So we're going to put an F. They're made of protein. They're used for attachment. They are found on gram-negative cells. That is true. They are composed of pilin. The last two that we have are they are composed of pilin, and they may be used for motility. Well, we already said that it's made for adhesion. It's used for adhesion. So we know that they are composed of pilin. So the answer, they are not used for motility. That is the correct answer. They are not used for motility. They are used for adhesion. 7. Which of the following pairs is mismatch? Glycocalyx for adherence. That is true. B. Pilin for reproduction. Well, pilin is used to transfer DNA from one cell to the other, and it's also used for motility. We have C. Cell wall and toxin. Well, some of the components for the cell are on the cell wall can be used as toxins, or be considered as toxins. Cell wall protection. Well, the cell wall protects the bacterium. And E. Plasma membrane transports. Well, there are transporter proteins on the within the plasma membrane that allows large molecules or a little smaller molecules to enter inside the cell from higher concentration to lower concentration. So we have true for this one, true for this one, and true for that one. So what we're left is with B. The answer is B, because I said that pili are used to transfer DNA information from one cell to the other, and it's used for motility, not for reproduction.